Good morning, dear students. We welcome you for today's online session for the content on the bryophytes, where we will be discussing about the type study of marchantia. If you see this marchantia, which belongs to the class Hepaticopsida, and the order Marchantiaceae and the family Marchantiaceae. So this marchantia is a thalite plant with 65 species worldwide distributed, mostly grown in the cool, moist and shady places, and Marchantia polymorpha grows in the burnt forest soils after fire. In India, 11 species have been reported. In Himalayas and South India, Marchantia palmata is found in eastern Himalayas, and the reproductive phases of this Marchantia are found during February and March in the Himalayas and during October and November in the South Indian hills. Regarding the vegetative structure, the external features, when you see the vegetative plant is a hematophytic plant body, which is prostrate, dorsiventral, dichotomously branched, dichotomously branched, with having a distinct midrib on the dorsal surface with a shallow groove, and on the ventral surface are present the rhizoids and the scales. The rhizoids are unicellular and hyaline, and the scales are multicellular and violet in color. Gamma cups contain specialized multicellular asexual reproductive bodies called the gamete. If you see the thallus internal organization, the anatomical organization of the thallus of marchantia, it is differentiated into three regions. One is known as the epidermal region, the photosynthetic region, and the storage region. The epidermis consists of the upper and the lower epidermis. Numerous air pores are present on the upper epidermis, and rhizoids and scales are present on the lower epidermis. In the photosynthetic region, which is present just beneath to the upper epidermis, this photosynthetic region is branched or simple. Photosynthetic filaments are present when they contain the chlorophyll cells. Beneath the photosynthetic region is the storage region. It is compact zone of several cells which are polygonal without any intercellular spaces, which consist of the mucilage cells, and protein granules and oils. If you see the reproduction in marchantia, most of the marchantia reproduces vegetatively and sexually. The vegetative reproduction takes place by means of fragmentation, by means of gametophyte formation, and the, mm, by means of mm, the adventitious branches. When we talk about the fragmentation in this, when the thallus gets older, they die and eventually disorganize, and the death and the decay reaches dichotomy, and the young lobes become separated. Each of these grow into a new thallus. In case of adventitious branches, in some species, special adventitious branches arise from the ventral surface of the thallus, and they develop the adventitious branches from the stalk of the disc of female gametophyte. They get detached from the parent thallus, decay of the connecting tissue, and form the new plant. The third type of vegetative reproduction is known as the gamocup, which these are the bird-like structure produced in large number, which are detached and carried by wind, water, and grow in the new individuals in the new number. This gamete is a cup-shaped structure with fringed margins, and each contains many buds. Each stand on the short stalk and possess mucilage hairs. The gemma are detached from the parent plant due to absorption of water and carried out by water current, and each gemma eventually germinate into a new individual. The other type of the reproduction that takes place is known as the sexual reproduction, and if you see the reproductive structures, there are two types of the most of the thallus in Marchantia are dioecious, where the Male and the female reproductive glands are born on different thalluses. So, if most of the reproduction takes place by the gametes, these gametes are present on special stalk-like structures which are known as the gametophores. These stalk, when present on the antheridia, are known as anthridiophore, and when present on archegonia, are known as the archegoniophores. So anthridiophores, mostly they are long stalks of 1 to 3 centimeters, 8 lobed, and each lobe have a numerous minute cavities in the antheridial chambers embedded in the photosynthetic region, and each chamber contains the single antheridium. The archegonium is also 3 to 5 centimeter stalk and a terminal disc which has a star shape with 8 to 9 radiating rays, and each ray has a 12 to 14 archegonia embedded in the fertile pockets. This antheridia has antheridial chambers inside which the antheridial chambers open exteriorly by ostiole, and each chamber contains a single antheridium. This mature antheridium is globular with a head and a stalk, and the antheridium has single layered sterile jacket enclosing mass of androcytes which metamorphose into anthrocytes. So this is the structure of the antheridium. Then comes the comes the archegonia, which is found in uh, serrate fertile pockets on the ventral ridges of the radiating discs. On the ventral ridges of the radiating discs, the enclosed the their archegonium is enclosed in pericytium, and this archegonium is pendant attached to ventral ridges. It each has an inverted flower shape, structures with bases full and part called the venter, and then this is the venter and this is the neck. Inside the neck consists of six vertical rows of cells called the neck cells, and encloses in 4 to 8 neck canal cells. The tip of the neck consists of cover cells, 4 cover cells. So fertilizations once they mature, the anthracites metamorphose, and these metamorphosed anthracites will develop into biflagellated juice pores. They swim with the water and they fall onto the on the ventral on the archegonial cells where it enters through the neck canal cells, and inside the venter it is present the egg where the juice pore fuses with the zygote and divides and forms the zygote. This zygote later will divide and form what you call the sporophytic body, plant body, which we call it as the sporogonia. The sporophyte of marchantia is consists of foot, seta, and capsule. The zygote divides by horizontal wall at right angles to the other and leads to the formation of a epibasal and hypobasal region. The capsule is formed from the epibasal region. The hypobasal cell gives the foot and seta. The cells in the capsule by periclonal division gives rise to different layers of the capsule. The outer is known as the amphithecium, inner is known as the endothecium. The cells of the amphithecium undergo divisions to form the capsule wall. The amphithecium undergoes the capsule wall. The cells of the endothecium gives rise to archisporium, and archisporium matures and forms the spore mother cells, where the spore mother cells divide meiotically and form the spore tetrads, and few cells differentiate and elongate to form the elytex. And each mature spore, two spore coats, they have two spore coats. The outermost is known as the exine and the inner is known as the intine. So at maturity, the layers separate to release the spores. The young sporangium is protected by three sheaths, the periganum, the calyptra, and the pericytium. So this is the structure, the calyptra, the pericytium. This is the foot, the seta, and the capsule. If you see the life cycle, mostly it is it is heterologous type where the gametophyte is the dominant phase and the sporophyte is the sporangium, which is different. So it is a haplodiplontic type of the biontic type of the life cycle. So these are the heterothallic dice where the on this thallus are the male gametophyte and the female gametophyte present on the stalks called anthridiophore and archegoniophore, and on the ventral surfaces are present the anthridia and the archegonia. At the maturity, the raindrops will take the sperm onto the near to the archegonia neck where it enters, and once it enters, many sperms will enter, but only one will fuse and form the egg, and the zygote is formed. This zygote, which is a cell, so we call it as layer, it forms divide and form an embryo, and this embryo will later develops the young sporophyte, which is diploid, and that leads to food seta in the capsule, where the sporangium is deposited on the gametophyte, and then this meiosis, the spores in the sporangium, they will the meiotic division, and they will form the spores, and these spores will develop into what we call the protonema gametophytic structures, which later forms the rhizoid in the thallus.